This paper presents a systematic and comprehensive benchmark of 11 machine learning methods for predicting in vivo RNA protein interactions 87. The authors address a major challenge in the field, the lack of standardized evaluation, by testing these methods on 3 large, distinct KELIP-seq datasets using a unified data processing pipeline and evaluation framework 88. The study rigorously assesses the impact of key variables on model performance, including neural network architecture, input data types, e.g., sequence versus structure, and crucially, the strategy used for selecting negative training examples 8,989. I would classify this paper as a methodological benchmark that is also good for teaching. It doesn't present a single new finding or method, but rather provides a critical service to the field by systematically evaluating existing tools on a level playing field. This is invaluable for users who need to choose the best tool and for developers aiming to improve upon the state of the art. It's excellent for teaching because it clearly lays out the complexities and potential pitfalls of model evaluation in computational biology, such as the profound impact of negative sample selection and dataset-specific biases. The holy grail question here is a meta-scientific one. Among the dozens of computational methods developed to predict RBP binding, which ones actually work best and why? For the past decade, and especially with the rise of deep learning, there has been an explosion of new tools for this task. However, progress has been difficult to track. This connects to a long history in computational biology of bake-offs and benchmark challenges, like CASP for protein structure prediction and the DREAM challenges. In the early days of motif finding 20 to 30 years ago, comparing tools like MeMe and Gibbs Sampler was relatively straightforward. However, as methods grew more complex, incorporating diverse data types and using sophisticated machine learning, direct comparisons became nearly impossible. Each new paper would claim state-of-the-art performance, but would typically be evaluated on a different dataset, with different pre-processing and different negative controls, making it an apples-to-oranges comparison 90,909,090. Leaders in this meta-research space are those who take the time to perform these rigorous, often thankless, benchmark studies. This paper by Annalisa Marsico's group follows in that tradition. It addresses the critical need for standardization, allowing the field to move forward by building on a clear understanding of what architectural choices and input features genuinely drive predictive power, versus what might be an artifact of a biased evaluation setup. Okay, that's enough of the background. Now we're going to talk about the first figure, Figure 1. Figure 1 provides an overview of the field and a schematic of the benchmark study design. Figure 1a is a bar chart showing the number of published RBP binding prediction methods over time, categorized by their core architecture, CNN, RNN, GNN, etc., 91. It clearly illustrates the explosion of deep learning methods, particularly CNN-based ones, starting around 2017-2018. Figure 1b is the conceptual diagram for the entire study 92. On the left, it lists the 11 representative methods selected for the benchmark, grouped by their architectural class, e.g., CNN, RNN, language model, shallow learning. In the middle, it shows the 3 large CLIP-seq datasets used, Mukherjee PAR-CLIP, ENCODE-eCLIP, and IONMF93. A key aspect highlighted is the use of 2 different negative control sampling strategies, labeled -1 inch, unbound regions on expressed transcripts, and -2 inches, binding sites of other RBPs, 94. The right side outlines the 4 major questions the benchmark aims to answer. 1. Overall performance of different architectures. 2. The impact of the negative control choice. 3. The impact of cell type. And 4. The impact of input modalities, e.g., sequence versus structure, 95. Figure 2 presents the main performance results of the benchmark across all methods and datasets. Figure 2a shows the distribution of AUROC scores for the binary classification methods when trained using the -1 setting, random unbound regions as negatives, 96. We can see that most deep learning methods outperform the shallow learning method, GraphProut. Among the deep learning models, RNA-Prout appears to be the top performer in this setting, 97. Figure 2b shows the AUROC distributions for the -2 setting, binding sites of other RBPs as negatives, 98. This is a much harder and more realistic classification task. The multi-label models, DeepRipe, multi-resbind, multi-RBP, which were excluded from Panel A by design, are now included 99. In this more stringent setting, the multitask models, particularly DeepRipe and MultiResBind, emerge as the top performers, outperforming all single-task binary classifiers 100. Figure 2c directly compares the AUROC scores for each model in the -1 versus -2 setting 101. Almost all points fall below the diagonal, indicating a universal and significant drop in performance in the harder -2 setting. This strongly suggests that models trained in the -1 setting are learning to exploit experimental biases to distinguish bound from unbound regions, and this advantage disappears when discriminating between two sets of biased, bound regions 102 billion, 102 million, 102,000, 102,000, 102. Figure 2D and 2E are control experiments. The authors had to modify the original multi-label methods, DeepRipe and MultiResBind, to handle the -1 setting. These panels show that their modified versions perform slightly better than the original implementation, confirming that adding a universal negative class improves performance as expected 103. Figure 3 dissects the factors that contribute to model performance, focusing on input features and input size. Figure 3A and 3B investigate why RNA-prot performed so well in Figure 2. They compare the full RNA-prot model to a version trained on sequence-only, RNA-prot sequenly, 104. In both the -1 Panel A and -2 Panel B settings, the sequence-only version performs significantly worse. This demonstrates that RNA-prot's high performance is largely driven by its use of auxiliary inputs, namely sequence conservation scores and exon-intron annotations 105 billion, 105 million, 105,000, 105. Figure 3C and 3D explore the impact of input sequence length, using the Peister model as a test case 106. They compare the standard Peister model, which uses a 400 nucleotide input, to a version restricted to a 101 nucleotide input, PYSSTER 101. In both negative settings, the model with the larger input window performs substantially better, suggesting that a broader sequence context is important for prediction 107. Figure 3E, 3F, 3G, and 3H examine the contribution of RNA structure information 108 billion, 108 million, 108,000, 108. The panels compare structure-aware models, IDEEPs, which uses predicted structure, and PRISM-Net, which uses experimental IC shape data, against the average of sequence-only models. While on average there is no major benefit, the scatter plots reveal specific RBPs, highlighted in color, for which adding structure information does appear to boost performance, such as EWSR1, PUS1, and Caprin 1109. This suggests that the utility of structure information is RBP-specific. Figure 4 explores the influence of dataset properties, specifically cell type and experimental protocol, on model performance. Figure 4A and 4B plot each model's AUROC for a given RBP against the median AUROC for that RBP across all methods 110. The strong correlation shows that some RBPs are simply easier or harder to predict, likely due to the quality of the clip data or the inherent specificity of the RBP itself 111. Figure 4C shows that model performance generally increases with the size of the training dataset, i.e. the number of called peaks, as expected 112. Figure 4D compares the performance of models for the 73 RBPs that were assayed in both K562 and HepG2 cell lines 113. Performance is highly correlated, suggesting that for a given RBP, models perform similarly regardless of the cell line. Figure 4E and 4F address cross-cell type prediction. They plot the performance of a model trained and tested in the same cell line, x-axis, versus the same model trained in one cell line but tested on the other, y-axis, 114. There is a noticeable but modest drop in performance. Points are below the diagonal, indicating that models do learn some cell type-specific features, but still generalize reasonably well, 115,115. Figure 4G, 4H, and 4I perform the same analysis but for cross-protocol prediction, comparing models trained on ENCODE EKIL-IP data versus Mukherjee PAR clip data 116. Here the drop in performance is dramatic, panel H and I. A model trained on one CLIP protocol performs very poorly when tested on data from another protocol, suggesting that protocol-specific biases are very strong and are heavily learned by the models 117,117. The main conclusions from this comprehensive benchmark are: Multitask models outperform single-task models in the most realistic evaluation setting, likely because they learn to ignore shared experimental biases. 118. Input features matter more than architecture. Adding sequence conservation and genomic context provides a major performance boost, as does using a larger input sequence window, 119 billion, 119 million, 119,119. There was no clear winner among the deep learning architectures. CNN versus RNN, themselves 120. Negative sample selection is critical. Models trained to distinguish bound sites from random unbound regions achieve inflated performance scores, likely by learning C-ellipsec artifacts. A more rigorous evaluation uses the binding sites of other RBPs as negative controls. 121. Models generalize reasonably well across cell types, but fail to generalize across different C-ellipsec protocols, highlighting the powerful influence of protocol-specific biases. 122 billion, 122 million, 122,000, 122. The implication is that future development of RBP prediction tools should focus on multitask learning frameworks and the incorporation of rich, informative features. Furthermore, the community must adopt more rigorous and standardized evaluation procedures, particularly regarding the choice of negative controls, to ensure that reported performance gains are real and not artifactual. The big picture objective of this study was to bring clarity and rigor to the field of computational RBP binding prediction by creating a standardized framework to systematically benchmark existing methods and identify the key drivers of performance. 123 billion, 123 million, 123,000, 123. The paper's data fully supports its conclusions. The head-to-head comparison under a unified framework provides a clear ranking of methods and reveals that design choices like multitask learning and feature-rich inputs are more important than the specific type of neural network used. The stark difference between the negative sampling schemes and the cross-protocol evaluation provides a crucial, cautionary tale for the field about the dangers of biased evaluation and the pervasive influence of experimental artifacts. This work provides an invaluable resource and a clear guide for the future development and assessment of RBP binding prediction tools.